The title of my article is Gel Protects Monkeys from HIV After Sex. Study finds. The author is Donald J. McNeil and it was published on March 12, 2014. It seems that the purpose of the article is basically to enlighten readers on the different research findings on the prevention of the HIV infection. Now, these research findings have been mostly done on monkeys. Actually, they all have been done on monkeys. However, it seems that he wants to convey that we are getting closer and closer to finding a successful preventative method against the HIV virus in humans. But there does seem to be a little bit of controversy over the testing of the HIV prevention methods because of the uncertainty of who will be the first to try it. How will the placebo be administered, or what will they use as a placebo? And does the success in the trials with monkeys predict the success in human trials? I learned that there are actually a number of drugs that have been shown to be pretty successful in monkey trials. For instance, one specifically is an integrase inhibitor vaginal gel. It works instantly once it's applied, and it basically just fights against the virus. But the way it works is it prevents the virus from integrating itself into DNA of an infected cell. Another interesting fact was 2 American babies were actually cured of the HIV infection, but they were given a mixture of drugs soon after birth. If I were to further research this topic, I would definitely look into the studies that have been done by Dr. Henine, the director of the primate trials of the HIV prevention methods for the Centers of Disease Control and Prevention. I could also research the findings that were done by the other researcher mentioned in the article. Her name was Sharon Hiller. She's the leader of the human trials of the vaginal microbicides. So they definitely eager in the prevention of the HIV infection, so I think they would be good resources to look into. To me, this article closely relates to the course because of the experimental theme that's mentioned throughout the article. It mentions researchers, research findings, certain trials, the difference between human participants and monkey subjects. But I guess to be a little bit more specific, to me it seems that the researchers are unsure of the results these drugs will have on humans. Now, it sounds like that's closely related to the extent of the external validity of their findings. Another thing is that in the article it mentions a trial that was done on 9 monkeys. It was actually split up into 2 parts. But again, it was a total of 9 monkeys, and 2 out of the 9 monkeys still became infected after they were exposed to the prevention gel. Now, the researchers were puzzled as to why they still became infected, and they could not pinpoint why they were infected. Now, I found that that is related to the extraneous variable to certain experiments. Now, they could have still become infected because of something else, maybe a certain predisposition that they had, or just something that was kind of unexplained and was not under control.